Hello
Hello
everyone.
everyone.
In
In
today's
today's
webinar, I would like to welcome Dr. Shailesh. Just a brief background about him. He did his master's in biotechnology from MS Baroda University, and then he joined IIT Bombay for his doctoral studies. In his doctoral studies, he worked on different viruses under the guidance of Professor Kiran Kondabage and published several high-impact research papers during his tenure. Following completion of his PhD, he joined Haystack Analytics as scientist, and here he is working on the development of NGS-based molecular biology diagnostic kits. So please welcome him for this webinar, where he will shed light on how NGS is better and an advanced method of detection compared to RT-PCR that we see in routine diagnostic testing. So, Dr. Shailesh, stage is yours.
Thank you, Alakesh, for the kind introduction. Good evening, all the attendees. Today, as Dr. Alakesh already described, we'll be discussing RT-PCR and next generation sequencing, that is in short NGS. As you all might be aware of, real-time PCR or real-time polymerase chain reaction, which is also called by other names such as RT-PCR or quantitative real-time PCR or quantitative PCR, all mean the same. That is, the amplification of DNA is detected in real time. However, before I begin with real-time PCR and how it is useful in day-to-day life, I would first like to mention about Carimulis. Of course, no discussion on molecular biology or PCR can be done without talking about Carimulis and his contribution. So Carimulis was one of the first people who recommended that amplification of PCR reaction or amplification of a genome or a target of a DNA segment can be done in vitro using repeated cycles of heat denaturation and using a specific set of primers. For this contribution, he was, of course, awarded the Nobel Prize in Chemistry in the year 1993. While Carimulis is fondly remembered even now, many people tend to forget the contributions of others who were also his colleagues or his competitors and went on to contribute a great deal to the field of molecular biology. One such contribution is from a colleague called as Russell Higuchi, who was also a colleague of Carimulis, who was given the credit for the invention of the real-time PCR. While Carimulis was getting the Nobel Prize in 1993, there were two seminal papers which were published in 1992 and 1993 by Russell Higuchi and his group, which described a method similar to what we call today as real-time PCR. So this method basically employed simultaneous amplification as well as detection of specific DNA sequences and monitored the amplification of these sequences in vitro in real time. And that is what came to be known as the real-time PCR. So if you look at the very first kind of setups that were used by Higuchi and his colleagues, used a simple thermal cycler, which we use even now. It was coupled with a camera on its top, which could image the thermal cycler's reaction in real time and capture videos. These videos were then sent to a controller, which would take pictures at specific time points, and then this data was analyzed. The middle panel shows a very interesting experiment that was done by Higuchi and his colleagues, wherein they varied the cycle number from 0 to 28 and the relative input target DNA from 0x to about 192x. From left to right of the screen, as well as from top to bottom, we see that as the cycle number increases, if the relative DNA concentration, input DNA concentration was higher, the fluorescence slightly increases over time, which basically shows that real-time measurement of DNA amplification was possible. They also showed a very early application of this technique of real-time PCR to detect sickle cell anemia. They used two set of primers, one on the left side of the tube in each panel, which was specific for the wild type allele, whereas the other one, which was specific for the sickle cell anemia affected allele. Now, in case of an homozygous individual, you would see that the primer with the wild type allele, which amplifies the wild type allele, shows very high fluorescence, whereas the one which is responsible for sickle cell anemia does not show any fluorescence at all. In case of an heterozygous individual, both the tubes, the wild type as well as the sickle cell anemia affected patient, both will show amplification as both the target genes are present. Whereas in case of an homozygous individual, where the wild type gene is completely absent and only the defective gene is present, only the second tube, which is targeted to the sickle cell anemia primer, sickle cell anemia target gene, only shows amplification. So from this, in 1992 and 1993, RT-PCR has come a very long way. That is, the principle still remains the same. That is, the real time, that is, a set of primers are used to amplify the DNA sequence, and this amplified DNA sequence during its amplification is measured in real time. From the initial use of ETBR or ethidium bromide, we have moved on to more specific dyes such as cybergreen. The cybergreen dye has a very high fluorescence when it is bound to a double-stranded DNA. So a cybergreen dye-based method works on the principle that there are two primers, a forward and a reverse primer, which targets a specific part of the gene, of the target gene. And then after amplification, the cybergreen dye binds to this amplified DNA, double-stranded DNA, and emits fluorescence. As cybergreen can bind to any double-stranded DNA, including primer dimers or any non-specific amplification that is happening, a modification was made which involved a third primer, which is called as a Tachman probe. In case of a Tachman probe, what happens is, in addition to the two primers, that is the forward and the reverse primer, there is also an internal primer, which is called as the probe. This probe has one end which is attached to a reporter molecule which has fluorescence, and the other end is attached to a quencher molecule. When both of them are close together, they are not bound to the target. What happens is the fluorescence is quenched by the quencher, and therefore there is no signal that is observed. But when during subsequent cycles of PCR, when a 5' to 3' exonuclease activity of the polymerase chews up the probe, we start to see fluorescence. And this fluorescence can then be determined and separated from the background fluorescence by something called as a CT value, or the cycle threshold value. The CT value is basically a determinant of the amount of DNA that was present, and it is inversely proportional to the initial DNA input amount. Real-time PCR, because of its ease of use, has been used for many different applications. These include mostly in research about mRNA and mRNA gene expression, which is done by quantifying the amount of mRNA for a target gene. It is also used in SNP genotyping, that is single nucleotide polymorphism detection, or copy number variation analysis, which are slightly complex assays as compared to mRNA analysis. They are also used for measurement of mRNA and miRNA and long non-coding RNAs, which affect gene expression. And finally, they are also used for detection of mutations in genes as well as for microbe detection. So right from its application from the first commercial systems that were available, RT-PCR has been used widely for a number of applications which are listed over here. But the important event, the two important events that where RT-PCR really came of age are these two applications. The first one, which being the tuberculosis detection test, which is done using the GeneXpert, which also gives us rifampicin resistance, which was deployed from 2005 to 2010, and now has become a mainstream test for diagnosis of tuberculosis. And the other one was the detection of SARS-CoV-2 during the COVID-19 pandemic. As all of us must be aware, during this COVID-19 pandemic, RT-PCR became a household name, and everybody understood what was the meaning of RT-PCR as well as what was CT values, right? And this could happen because the test was rapid and affordable, and although it gave only qualitative results as opposed to its name, it was still effective in diagnosis of the infectious agent. Despite all of these advantages, RT-PCR comes with its own set of limitations. That is because it is only a panel-based test, and you can only target a certain number of targets in a given test. So right now, most of these RT-PCR tests would only target up to 5 genes or 5 targets in a single test. It is highly dependent on specific primers. Therefore, you require sequence level information, and unless a high quality sequence level information is available, there would not be any amplification or primer designing that can happen. It also targets only a small portion of the target gene. So those of you who have done RT-PCR would realize that RT-PCR amplicons are only 150 to 200 base pair long, and therefore they only target a small portion of a bigger gene, and therefore the target is very small, and this is a major disadvantage. Also, RT-PCR can only be used for detection of what is known, and therefore it has zero discovery power. If there is an unknown organism which is present in the same reaction, it will not be detected using RT-PCR. We'll talk about some of these examples in the upcoming slides. And that is where the limitations of RT-PCR have to be substituted with something called as a next generation sequencing. Now, next generation sequencing, or NGS, has been around for about 2 decades, and there have been several landmarks during these landmark events during these 2 decades. Right from 1975, when Frederick Sanger, who also won the Nobel Prize for showing sequencing in vitro, to pyrosequencing, automated Sanger sequencers, and then ultimately second and third generation sequencers, the DNA sequencing technologies have indeed come a long way. The major difference between the second and third generation sequencers, which are now often used in real life scenarios, is that the second generation sequencers are mainly short read. That is, they produce reads which are about 150 to 300 base pair long. Whereas the long read sequencers such as PacBio and Nanopore can can produce reads which are more than 1 kb or sometimes even 1 MB in size, and which helps in several different aspects, such as one being the sequencing of the human genome. Although the draft of the human genome was available in the early 2000s, it was not possible to map each and every, or to have sequence of each and every base pair which is present in the genome, mainly because of the complexity of the genome at the ends. That is the telomeric regions, which cannot be sequenced very easily using Illumina or ion torrent sequencing. Whereas long read sequencing, such as nanopore sequencing, is now being used to fill in these gaps which are present in our known knowledge, and hopefully will help us to get the entire genomic sequence of not just humans but all the organisms. Any NGS technology works in these four basic steps. That is, first, it requires a extraction of very high quality genomic DNA or RNA, whichever is the target that we are looking for. Second step involves library preparation, wherein the DNA that is obtained, or the RNA that is obtained, is prepared for its binding to the sequencer. So sequencer flow cells are specific and vary from technology to technology that is being used. Therefore, they need to be modified. The DNA needs to be modified in a certain way at its ends so that it can be detected by the sequencer. Once it is loaded onto the sequencer, the real-time sequencing happens, and after sequencing, the raw data is available, which then has to be bioinformatically analyzed and interpreted according to the experiment that was being done. I'll just briefly tell you about two sequencing platforms, that is, one being Illumina and the other being Nanopore. Illumina, as I mentioned earlier, is a short read sequencing platform, whereas Nanopore sequencing is a long read sequencing platform. In Illumina, works by a principle called as sequencing by synthesis. That is, the DNA that is to be sequenced is first attached to adapters. This adapter attached DNA is then loaded on a flow cell, where it forms something called as a bridge amplification, and synthesis of DNA occurs. A polymerase enzyme actually extends the DNA, and every time a new nucleotide is incorporated, a fluorescent signal is emitted. This fluorescent signal, which is formed in clusters, is called as cluster formation, and this fluorescent signal is later on analyzed by high throughput cameras and sequencing and computers. Ultimately, it gives a raw read sequence, which then has to be further QC'd and used further for either assembly or assembly of the genome, or any other purpose that was the question in question. In nanopore sequencing, however, what happens is the mechanism of working of nanopore sequences is completely different, and it does not undergo sequencing by synthesis. What I mean by that is, a target DNA which has to be sequence can just be modified at its ends and loaded onto the sequencer. Inside the sequencer, there is a nanopore, which is a small pore which allows selective passage of DNA, and above that is a motor protein. The motor protein basically unzips the DNA from its double-stranded form to its single-stranded form and allows passage of the DNA through the nanopore. Once the DNA passes, as every nucleotide passes through this membrane, electrical signal change is formed. This is detected by the sensor chip, and the real-time DNA sequence can be obtained. A major advantage of nanopore sequencing over Illumina sequencing is that nanopore sequencing is cheaper. It can do native DNA sequencing or native RNA sequencing. It can also start producing data within 20 minutes of starting the sequencing run. And what that means is you do not have to wait for long hours before you start to get data. So it is a real-time sequencing, and because of its size, which is of the size of a mobile phone, it is highly portable and cost-effective, and therefore can be taken to any remote part of the world for sequencing applications. Now, NGS, because NGS is such a high throughput technique, it has been used for several different applications since its invention. One of the main applications being the gene expression through RNA-seq. Now, like we already mentioned, for NGS, for RT-PCR does mRNA analysis based on the expression levels of the gene. Whereas here with NGS, we can do a whole throughout expression profiling of the entire cell. So what advantage that it gives is, instead of looking at target genes of three or four target genes, we can look at the expression profile of the entire gene, entire organism, or the entire cell. NGS has also now been used for whole genome sequencing and assembly of new genomes. A fantastic example of this is the COVID-19 pandemic. Again, when the genome sequence of SARS-CoV-2 was deposited very early on during the pandemic, and this basically helped. This high-quality genome sequence that was deposited by researchers basically helped in development of RT-PCR test as well as other tests. It also helped in the manufacturing ultimately of the mRNA vaccine that is now available. Thirdly, it can also be used for whole exome sequencing, which is limited right now to humans, and which can tell us the impact of our genes on our overall health. And lastly, it can be also used for metagenomic direct or targeted sequencing to identify either pathogens or microbiome in an environment. So, because of the advantages that NGS provides, there are several cases in which NGS will always be better than RT-PCR. And one of the best examples that can be given is the spike gene target failure, which was observed during COVID-19. So during the COVID-19 pandemic, several RT-PCR tests from different manufacturers failed to identify the spike gene. One such example was the TACPath COVID-19 combo kit, which came from Thermo Scientific. When this test basically tested for 3 target genes: the nucleocapsid gene, which is the N gene, the S gene, which is the spike gene, and the ORF1ab gene. Now, because spike gene undergoes a lot of variations during the growth of the virus, it tends to keep changing throughout the evolutionary phase of the virus. So what was observed was the N gene, that is a nucleocapsid gene, and the ORF1ab gene could produce amplification using this kit, but the S gene amplification always failed for some samples. When these samples were sequenced or were put on whole genome sequencing, it was identified that these are the 2 different variants of coronavirus. That is the alpha variant, which was the very first variant of concern that started to come in, and later on the Omicron variants. Now, both of them show a deletion of 2 amino acids at a specific position that does not allow the amplification using the S gene. This S gene deletion is not seen in Delta variant, and therefore this Delta variant could be detected by RT-PCR. Now, what this basically concludes is that because RT-PCR is so specific to a small portion of the genome, if that portion undergoes some modification or is deleted, then the test will basically give you a false negative report. This is a graphical representation of what I just showed you. Now, this shows that the positive predictive value of S gene target failure sequences was very high during the rise of the alpha variant, which is shown here in pink, and again during the Omicron variant, where it clearly shows that there is a rise in S gene target failure using RT-PCR kits, because these 2 variants do not show a small deletion within the S gene. A second case is again very interesting, and this comes from our own experience, where we were trying to test a nucleic acid extraction kit from a commercial vendor, wherein this commercial vendor had been using these kits for RT-PCR based diagnosis. And when we extracted 3 DNA samples, which are shown here in A, B, and C, D and E were other DNA samples which were extracted using another kit which served as a control. What we could observe was all the 3 samples which were extracted, the DNA was extracted using the kit A, showed the presence of 3 organisms in high numbers. That is this Novospingium humi, Curvibacter, as well as Acidovorax. Whereas these organisms were not present in the control DNA that we had extracted using our own kit. Now, what this basically shows is that RT-PCR, while this kit was working perfectly fine for RT-PCR, when it came down to sequencing, the real life scenario was seen. These kits were probably contaminated with these 3 organisms all throughout. But since RT-PCR did not target any of these 3 organisms, it could get away with it. But when we put it on sequencing, when we put it on Oxford Nanopore sequencer, we could see that very easily these 3 organisms were present in all the samples extracted using these kits. A second example, the third case is again related, wherein we used a commercial automated DNA extractor from commercial vendor B and sequenced the DNA obtained on an Oxford Nanopore sequencer. What we could see was there was a 16S amplification in all the samples, which made us happy, thinking that the amplification shows that there is presence of organism. But upon further analysis, we found that in addition to the expected pathogen, which gave the 16S amplification, there was also this pathogen, which is this organism called as Pingomonas lady, which was present in all the samples in low abundance, which again shows that there is a kit level issue or kit level contamination that is present, which is being missed on RT-PCR, but it's clearly visible on a NGS based platform. The last and final example of RT-PCR of NGS being better than RT-PCR is that it allows the discovery power, or it allows us to detect everything that is present inside a sample. So in this case, our team from various centers could detect sequencing on Oxford Nanopore could detect some rare pathogens which would have been missed had we done an RT-PCR panel. And some of these organisms include Elizabeth kingia, Orientia susugamushi, and Rickettsia, both of which cause typhus fever, and other pathogens with their resistance profile. Now, had we put these samples or this DNA on an RT-PCR test, which only detects a certain number of organisms, or a panel-based organisms of 20 to 25 organisms, it would have failed to detect any of these organisms. But since we did whole targeted sequencing for these samples, we were able to detect all three of them very easily. So to conclude, basically a comparison of RT-PCR and NGS is very easy, because NGS is a much superior technique as compared to RT-PCR in the current times. So RT-PCR, while it is panel based and hence it detects only a number of targets, few number of targets, NGS can give high throughput data, and therefore it can determine unlimited number of targets that can be screened at a single time. RT-PCR is highly dependent on designing primers, and therefore it is dependent on sequence information that should be available from genome sequencing platforms. Whereas NGS does not really depend on specific primers. It can work independent of primers if you use it in the whole genome, if you use it for whole genome sequencing. But of course, if we have to use it for targeted sequencing, that is, you want to only target a certain gene within an organism, then specific primers would be required. RT-PCR only targets a small portion of the target gene, so it only amplifies about 150 to 200 base pairs in a gene, or from sometimes even from an entire genome. Whereas NGS is capable of collecting information from as low as few bases to the entire gene or even the genome, and therefore it gives us a lot of other information that might be useful. RT-PCR again has zero discovery power, because it only detects the known. Whatever organisms or panels or primers have been designed, it will only detect those. Whereas NGS is a fantastic tool for new pathogen discovery along with its routine testing applications. So thank you. Lakesh, I think you need to unmute.
Oh, sorry. So it was a really nice talk, Dr. Shailesh. So I think this NGS-based technology will definitely revolutionize the way we think clinical microbiology. So I guess we're open for Q&A session. So audience, please, you can please ask anything related to NGS or RT-PCR. Okay, we got one question. So between targeted and total nucleic acid NGS, which is better and why?
Okay, so this question actually the answer to this question depends on what is the application that you're looking at. So targeted NGS can be very good in when we are actually dealing with clinical samples. So for instance, from our own experience, what we have seen is when we try to do whole genome sequencing, non-targeted for clinical samples, what we end up sequencing is a lot of human DNA. Because human DNA will be a part of the body samples body or fluid samples that we take for sequencing. So there will be a lot of human DNA that will be sequenced, which we don't really need for our analysis because it is host DNA and non-pathogenic. It will also give you so there will be a lot of host DNA that will be sequenced if you do whole genome in that case. But if you do a targeted sequencing, that is, if you are only looking for, let's say, bacteria or fungi or a virus, then it would give you much better yield, much better confidence to call that organism. So there have been cases where we have subjected the same sample to targeted NGS using 16S or ITS amplification, and we have also done the same sample on whole genome sequencing. And what we end up seeing is in whole genome sequencing, although it is more sensitive, we tend to get a lot more host DNA, which is not really helpful in diagnosis. But in the case of targeted amplification, we start to see identification of organisms as well as antibiotic resistance that will be more useful in treatment of the patient. So yeah, so in one line, it basically depends on what is the question that you're asking.
And which is more cost effective? What do you think?
So cost effective would be obviously targeted. Targeted sequencing will be cost effective because the amount of data that you require in that case will be lesser. Also, it will be better in terms of the turnaround time that we can give. So for instance, nanopore sequencing that we use for the infection test, it gives a turnaround time of about 12 to 13 hours, which helps us in detecting detecting the infection timely. So nanopore based sequencing technologies are obviously more cost effective as compared to the whole genome based kits or whole genome based applications.
So I have one question, Dr. Shailesh. So we all know when we do sequencing, there comes lots of data, and there requires lots of bioinformatic analysis. So
Right.
as an audience, what a person should do once they get this data?
Okay, so the information that is available in or rather that the information that you get from a sequencing platform, there will be a lot of noise as well in that. So the first step would be QCing, determining which sequences are worth taking forward for analysis. So that is one step of it. Obviously, you will need some bioinformatics skills in order to do that. But right now, there will be some tools available online which can do that. We of course offer, as Haystack Analytics offers certain tools which you can use in order to analyze this data in a completely automated manner as well. So yeah, so there are different things that you can do with it depending on what you want to find out.
Okay, thanks. So is there any more questions from the audience? Okay, I think again I would like to thank Dr. Shailesh for giving such a nice this NGS versus RT-PCR is something everyone should look into this and understand why people should opt for NGS-based diagnostics. So thanks again. Thanks. I want to thank all the audience for attending this webinar. So hope we will see you guys again. Yeah. Bye.
Thank you.
Bye.